Everybody. My name is Bradford, and today I want to show you how to use the Triple 3 Biosample Loader. So this is a nearly empty triple site that I've kind of built for this purpose. You can see if I go to Content, Triple Content, we have about 200 mRNAs. I have genes in the database, but I don't have them published as entities. So right now I have mRNAs, and I have one organism, Fractionis excelsior, which is what we're going to load it for. So the goal is I have this expression data, and I need to first I need to create biomaterials, and or biological samples, and then for each think of that as that a biological sample is a given tissue sample. It's a given RNA extraction run. It's some treatment that you made that you've collected sample from, you've extracted RNA, and now you want to know the expression values of the genes in that treatment. So we're going to create biosamples, and then we're going to associate expression values for each biosample with each of the mRNAs that we have loaded in the database. So to get started, we click on Triple in the admin menu, and it's going to be in Data Loaders. If you don't see it in your data loader, then you have not enabled the biomaterial module. So you need to double check that that's enabled. So if I go, you can see I have Tripal Analysis Expression is the module that's responsible for loading biomaterials. We need to, and you want to be on the master branch. So we're going to check out the master branch. Oops, we're going to get pulled to make sure we're up to date. You can see a whole bunch of stuff has changed. Now we're on the master branch. So then we're going to use Drush to enable the module. Drush PM enable Tripal Analysis Biomaterial. And it's Tripal Analysis Expression, not Tripal Analysis Biomaterial, so let's fix that. I don't know why my text is wrapping kind of weird there. Sorry about that. So if we want to continue, we say yes. Okay, so now that the module is enabled, we see the Chado biological sample or biomaterial loader shows up in the data loader section of the admin page. So we click on it. First, we need to provide a biosample file. So these you can download from NCBI. If you have a list of NCBI biosamples, you can produce an XML formatted file. So we can upload it. And you can associate it with an analysis. I haven't created one to associate it with, but there's, that's easy to do. There's other material I have for that. And we need to select an organism. So what happens here is kind of interesting. We can just go ahead and import it, but for Triple 3, we've added all sorts of controlled vocabulary integration. So if you press this Check Biosamples button, what it's going to do is it's going to read the XML file, and it's going to check the term as well as the value for the term and see if there's a controlled vocabulary term that matches it. And if so, it's going to let you associate it with it. If not, it's going to insert it. By default, it's going to insert it for the property. It's just add it to. You can see here, I already have tissue as a biomaterial property in the controlled vocabulary, which is essentially a made-up controlled vocabulary that's just going to exist on a triple site. So it's not ideal. It works. And then for the value, it's going to be if you don't associate, it's going to associate it as free text. So that probably doesn't make a lot of sense. So let's take an example. So I can see here that one of my biomaterials, or one or multiple of my biomaterials, uses tissue. So the tissue for this biomaterial, it might have been the tissue is the property, and the value is going to be leaf, right? It was a leaf tissue sample. So I can say, yeah, you can load it in using this vocabulary. If I'm not happy, maybe I want tissue to use the plant trait ontology term, I would load in the plant trait ontology control vocabulary and make sure that there is a term with that's the name is tissue. If let's say this file had a typo and it's misspelled, I would need to correct the input file so that's going to match the term that's loaded in the vocabulary. So we can keep that as is. Here you can see we have this vector replicon property, which matches in a term that's already in our database, the sequence ontology. So if I don't press anything, it will load in the property as vector replicon in this ontology, in the biomaterial property. But I would probably prefer to use the sequence ontology. So if I check this box, it'll load it in that way. Now it's telling me that there was this property, ENA first public, and I don't have a term that matches it. I could go back and add a vocabulary so that I have a term that matches ENA first public, or I can leave it as is and it'll get loaded into the generic biomaterial property vocabulary. So I'm going to go ahead and just continue to use the generic ones. So those are the property, right? The tissue type. And then you can see here I have organism part, and it's also going to get loaded as tissue, which is what we want. We don't want multiple terms using the same thing. So here you can see these are the values associated with those given properties, right? So tissue, the value of a given tissue might be quiescent and swelling buds. Or this 10 probably goes with, I don't know what it goes with. Let's say it goes with probably one of these, ENA first public. So, again, if I had a term in my vocabulary that matched any of these, it would show up here. Let's try loading the, if we loaded ontology, they would show up. So anyway, I'm going to do it as is like this. So these are going to go in the free text value. So you just press import biosample. It creates a job. You can just copy this. We're going to run the job in the terminal, and hopefully this runs with no errors. Awesome, it does. So I don't actually recall if it creates the content for us. So if we reset, do we have biological samples now? We don't, so we need to publish them. So to do that, we click Publish Triple Content. So what does this mean? It means that the biosamples are in Chado. They're in the database, but Drupal doesn't know about them. So if we go to biological sample, we press Publish, and we run this job, it should find those new Chado records that don't have a corresponding entity and create entities for them. And you can see that's exactly what happens. It found 3 samples, biological samples, and published them. So now if I go back to content, triple content, I now have these 3 biosamples. And I can look at them. And interestingly enough, you'll see that the properties don't show up out of the box for these biosamples, just the name and the description. So if let's say I want the properties to be a field, to show up as fields here, I need to configure the fields of this content type of the biological sample. So to do that, you go to you go to Structure, Triple Content Types. And here under biological sample, I need to manage fields. So this lists all the fields that are currently configured. Each of those properties will need to be its own field. So let's take a look at how that happens. if we just click check for new fields, it should get auto-discovered. Might take a second. Okay, so after pressing the check for new fields button, you see this message that it found a bunch of new fields, most of which are triple. So the way these work, right, is it's going to be the DB, what Chado calls the DB, or the DB short, what we're calling the DB short name, and the accession. So, or I'm sorry, and the term name. So triple, last update is one of the got inserted into the triple biosample vocabulary when we imported the biosample file. Versus it created this field SO vector replicon because we had a term for that in the sequence ontology. In every case, all those properties got added as terms. Got added auto-created as fields, as these Chado property fields. So if we actually go back to the biosample, we still won't see the properties displayed, and that's because the fields got created, but they don't display by default. We have to scroll down, and you can see them. They're all listed here under disabled. So what I typically want to do is we'll make a new group that's going to be like properties. Enable it, and we got to name it. Oops. Properties. And we want it to be a triple pane so that it matches. You can see the summary table, the relationship, crop references. These are triple panes. And then let's go ahead and we'll stick. Oops. You might find this. Honestly, this is kind of an annoying thing where dragging them around, you might have issues with that. I always do. So first, let's save the new group, and then we'll enable it, and then we will drag the new properties into that group to enable it. A few seconds, and now you see we are brought back, and you see that the group was created. Where is it? Yeah, even though I dragged it up to be enabled, it still was. It is disabled when it gets created. So then let's just drag all the properties in here so that they're part of this group. And of course, the order matters. That's how it'll be displayed to the user. So you might want to, because that odd bug, you might want to put some thought into how these get ordered. And then we'll just switch it from disabled to enabled, and all the fields that were in it were there. And so now it's, again, the order matters, right? So it's going to be listed under the summary table. So let's save this configuration. This will take a few seconds, and then let's visit a property and see how it looks. Okay, now that our settings have been saved, we can go ahead. We can go back to set of structure. Let's go. Now we should be set. So if we visit a biosample page, these fields should be enabled. So now you can see about the property and the biomaterial, and now we have this properties group, which we click on right now. I think something's not quite right with the display, but you can see that the property, now we have the property show up here. We actually, in this case, we really want the label. Right, this one is the tissue type is what the label is, and we really need the label for this to make any sense. So we should go back and configure it to look a little bit like this. What happens if we do that? We go to structure, triple content types, manage display, and then so if we look here, you can see it says the organism, these things appear in line. This is the label. So let's switch these to appear in line. And then also, let's think a bit. You see how here. Well, let's save this and see how it looks, and then so now let's look at properties. So this is a little bit better, but I think what we really want to do is have it show up in a nice table like this. So to do that, let's go one final time, go back to structure, triple content types, and just hit display, and you see that actually the triple pane also has a table inside of it. So what happens if we create a table? So we'll call it our Chado properties table. Chado table. And we'll try enabling it. It'll probably auto-disable once it's created. Okay, so now we've got the table created. So let's craft a little. What we want to do is we want to stick it right up in the in this triple pane we created. I'm kind of just swiping it up. There we go. So we want it in here. Now we want everything to be inside of this table. So we push everything, indent it all down. Like so. Let's save. Okay, so it loads. Well, let's take a look at our properties. All right. We probably want to get rid of this label, but then we probably want to capitalize this so it matches these other groups. But now we have each of our properties in this nice little table using mostly just the interface. So thank you for watching. I hope this was helpful for understanding how to load biosamples in 333. Probably what you'll want to do next is load the expression data associated with the biosamples, and I'm going to cover that in a separate video. So I'll see you then. Thanks.